Welcome to the deep dive. We're here to take a whole stack of sources and really boil them down to what you need to know fast. Today, we're jumping into something that honestly sounds like pure science fiction, but it's becoming very real. It's called mirror life. Just imagine for a second that everything about life as we know it, our DNA, the proteins doing all the work, imagine it had an exact opposite mirror image.
Okay.
And what if that mirror image could also be alive? It's a wild idea, right? But it comes from something pretty fundamental, the handedness of molecules, chirality. Now, this offers some incredible possibilities for medicine therapies, but it also carries this, well, unprecedented risk, a potentially catastrophic risk if it ever learned to replicate itself.
Yeah, and that's exactly what we're diving into today. Our mission here is to trace this idea from its, historical scientific roots,
Yeah.
all the way through the latest breakthroughs happening in labs right now, and then really explore the huge implications for health, for the planet, even for how we think about life elsewhere. We're going to dig into that tension between incredible promise and this really profound peril. And just so you know where we're coming from, we're drawing on a big report called Mirror Life, a comprehensive inquiry, plus a bunch of scientific papers, expert analyses, and notably a recent policy piece in Science from, I think it was 38 leading scientists weighing in. So a lot of heavy thinking has gone into this.
Okay, so to really get our heads around mirror life, we need to start with the basics of our life, this whole handedness thing. What exactly is chirality?
Right. So chirality comes from the Greek word chair, meaning hand. And it's basically about asymmetry. Think about your hands. They're mirror images, right? But you can't put a left glove on your right hand. They aren't superimposable. Molecules can be the same way. These two non-superimposable mirror image forms are called enantiomers. Now, chemically, they might look the same on paper, same melting point, boiling point, all that. But their 3D shape, their handedness, means they interact totally differently with other chiral things, especially in biology.
That hand analogy really works. Chemically identical, but geometrically opposite. And that difference is everything in biology, isn't it? I remember reading about some striking examples, like how it affects smell.
Oh, absolutely. Carvone is the classic example. One enantiomer, one hand, smells exactly like spearmint. It's mirror image. Smells like caraway seeds. Totally different. And it's because those molecules fit differently into the chiral receptors in your nose, which are themselves made of handed molecules, proteins. But it goes way beyond smell. To see how critical this is, tragically, we just need to look back at thalidomide. Back in the 50s and 60s, it was sold as a mix, a 50.50 mix of both enantiomers. One hand was a perfectly safe sedative.
Okay.
But the other one, its mirror image, was a horrific teratogen, caused terrible birth defects, and a devastating lesson. In a biological system which is built with specific handedness, these mirror images act like completely different drugs, life and death differences.
Yeah, that really drives the point home. Which brings us to this big puzzle about life on Earth, this homochirality. Why does all life here seem to use the same hand?
It's honestly one of the biggest unsolved mysteries in science, our proteins, almost entirely made of left-handed amino acids, except glycine, which is acryl, no handedness, but our DNA and RNA, they use right-handed D sugars. And DNA itself twists into that famous right-handed double helix. This preference is universal. Everything alive uses the same standard. But there's no known fundamental reason why it had to be this way.
So it probably wasn't preordained. It was more like a frozen accident early on. Is that the thinking?
That's the leading hypothesis, yeah. Something in the early Earth environment, maybe polarized light from space, maybe chiral minerals acting as catalysts, gave one hand a slight edge. And then through replication and evolution, that initial tiny imbalance got locked in, like a cosmic coin flip that came up heads, and everything followed. And while this uniformity is amazing, letting complex systems evolve, it also creates this massive vulnerability, a universal blind spot.
Right. That quote from the report really stuck with me. If the entire biosphere is built with right-handed locks, it is defenseless against a left-handed key. That sets up the whole threat, doesn't it?
It perfectly captures the core risk. And, the person who first stumbled onto this whole concept, conceptually at least, was Louis Pasteur, way back in 1848. He was working with tartaric acid from winemaking, and he found something really odd.
Oh yeah, the Pasteur story is amazing. What was the puzzle he found?
Well, the natural tartaric acid rotated polarized light. It was optically active, but a synthetic version, chemically identical, called paratartaric acid, wasn't optically active. It did nothing to the light. He couldn't figure out why.
So how did he crack it? It involved tweezers, right?
Incredible patience, basically.
Yeah.
He looked at the crystals of the inactive stuff under a microscope, and he noticed there were actually two slightly different types of crystals mixed together. And crucially, they were mirror images of each other. So painstakingly, he physically separated them with tweezers into two piles, left-handed shapes and right-handed shapes.
Wow, that must have taken forever.
You bet. Then he did the key experiment. Dissolved each pile separately in water. One solution rotated light to the right, just like the natural acid. The other solution, from the mirror image crystals, rotated light to the left by exactly the same amount.
Uh-huh.
And when he mixed the two solutions back together, the optical activity canceled out, vanished. He realized the inactive stuff was a 50.50 mix, a racemic mixture of these two mirror image molecules, and this dissymmetry, as he called it, was molecular.
Incredible detective work. And didn't he also find something about how life interacts with these, something with mold?
He did, yes. A bit later, he grew a common mold, Penicillium glaucum, in that racemic tartrate solution. And he found the mold only ate the right-handed tartaric acid, the natural kind. It completely ignored the left-handed stuff, leaving it behind.
So the mold could tell the difference.
Exactly. It showed that life is stereospecific. Its machinery prefers one hand over the other. So Pasteur not only discovered molecular handedness, he showed that life cares about it.
Right.
And that observation, that biological selectivity, that was really the first hint, wasn't it? If life prefers one hand, maybe, just maybe, life could exist that prefers the other hand. A mirror life.
Fascinating. Okay, so that lays the groundwork. We understand handedness. Now, how do scientists actually start engineering a mirror world? Let's get into part 2. Engineering the mirror world. This seemed impossible for ages. What shifted?
What really changed the game was the emergence of synthetic biology. It's basically biology meets engineering. Designing and building new biological parts, devices, systems. There's this long-term ambition in the field, sometimes called the Build-a-Cell project, to create a completely synthetic, self-replicating cell from basic chemicals. Now, we're not there yet, even for natural life components. But the tools and knowledge being developed for that goal are exactly what you'd need to build a mirror cell. It provides the roadmap.
And wasn't there a huge milestone back in 2010 from Craig Venter's group? That seemed like a big step.
Oh, absolutely. A landmark achievement. They synthesized an entire bacterial genome chemically from scratch in the lab. Then they transplanted that synthetic genome into a recipient bacterial cell whose own DNA had been removed. And incredibly, the cell booted up using the synthetic DNA. It started making the proteins of the species whose genome they'd synthesized. It wasn't creating life from scratch, but it proved you could design and build the operating system, the genome, and have it run a living cell. A critical proof of principle.
Okay, so the operating system can be synthetic. Now, how do we get the mirror parts? Going from mirror molecules to actual mirror machinery, What were the early wins there?
Well, one of the first really significant breakthroughs was back in the 90s. Stephen Kent's lab managed to synthesize a mirror image version of the HIV protease enzyme. They built it entirely from right-handed D-amino acids.
No.
And the key finding, it was totally resistant to being broken down by natural proteases. Our body's protein-cutting enzymes couldn't touch it.
Ah, because of the chiral mismatch.
Exactly. That really sparked interest in using mirror molecules for therapies that would last longer. Then came progress with nucleic acids, synthesizing mirror DNA and RNA using left-handed L sugars. You can actually buy short strands of mirror DNA commercially now. And then, the really crucial part, enzymes. Creating mirror image polymerases, the enzymes that can actually read and copy mirror DNA.
That sounds like a major hurdle to overcome.
It was, and a really key milestone here was achieved by Dr. Tingzhu's team in China just in 2022. They built a working mirror image version of T7 RNA polymerase. This is a really common, important enzyme used in labs. Now, it was too big to synthesize directly.
Right.
So they did something clever. They synthesized it in 3 smaller mirror image pieces, or segments. And these segments were designed to then spontaneously fold and assemble themselves into the full functional mirror enzyme.
That's ingenious. Self-assembly.
Very much so. And then they used this mirror polymerase to do something remarkable. Transcribe the largest pieces of mirror RNA ever made. Specifically, the mirror versions of the ribosomal RNAs, the 5S, 16S, and 23S RNAs. These RNAs form the structural and catalytic core of the ribosome itself.
Okay, this is getting serious. But it also really throws that dual-use problem into sharp relief, doesn't it? Like the same tech that helps make a mirror enzyme for a vaccine. It also makes it easier for someone else to build the core of a mirror ribosome, potentially for more dangerous reasons. How do you even govern that?
That's precisely the dilemma. It's a scaffolding of enabling technologies. Every advance, even if intended for good, lowers the technical barrier to eventually creating a self-replicating mirror organism. It makes governance incredibly tricky.
Which leads us right to the biggest challenge of all, building that mirror ribosome and then the whole self-replicating cell. Why is the ribosome considered such a monster challenge?
Yeah, people call it the ultimate bottleneck. It's arguably the most complex piece of machinery in the cell. Its job is translating the genetic code from mRNA into proteins, the cell's protein factory. And it's this incredibly intricate structure made of several large RNA molecules. Those rRNAs Ting-Zhu made mirror versions of plus dozens, usually 50 or more, distinct proteins, all folded and packed together perfectly.
So Zhu's mirror RNA work was huge, covering maybe two-thirds of the ribosome's mass. But getting those 50-plus mirror proteins synthesized and getting them to assemble correctly with the mirror rRNA, that sounds astronomically hard.
It is. That's the major remaining hurdle. Synthesizing all those amino acid proteins and then getting them to self-assemble with the L-sugar RNAs into a functional mirror ribosome that can actually churn out proteins. That's often called the sound barrier of mirror life synthesis. If someone achieves that, it's a game changer, moving from just making parts to having the core factory. And even then, you have the boot up problem, getting all the mirror components, the mirror genome, mirror ribosomes, mirror enzymes, all packaged inside some kind of membrane, and then kicking it all off so it sustains itself, replicates, metabolizes. That's a huge challenge, even for scientists trying to build synthetic cells with natural chirality. We haven't solved that yet either.
Okay, let's talk practicality. Timelines, costs. This sounds like it must be incredibly expensive and still pretty far off.
Oh, the practical hurdles are definitely immense. Just synthesizing a normal bacterial genome costs millions. The precursor molecules for mere DNA, those ill nucleotides, are way more expensive right now than standard D nucleotides. So the general consensus among most scientists is that creating a viable, self-replicating mirror bacterium is probably, at least a decade away, maybe more like 15 to 30 years, and it would likely cost hundreds of millions of dollars.
That sounds reassuringly far off.
Well, mostly. But you do have some leading figures like the geneticist George Church at Harvard, who's been much more aggressive in his estimates. He suggested it could potentially be achieved much faster, maybe even less than a year under certain circumstances.
Wow, less than a year. That's a huge difference.
It really highlights the uncertainty, right?
Right.
Predicting the pace of these kinds of breakthroughs is notoriously difficult. Someone could have a clever shortcut tomorrow.
Okay, that uncertainty really changes the feel of this. So we've seen how the engineering is progressing, the pieces are coming together. Now we absolutely have to confront that central paradox. The very properties that make mirror molecules useful are what could make a mirror organism so incredibly dangerous. Let's get into part 3, the double-edged sword. What's the core benefit again of the non-replicating mirror stuff?
Right. The core benefit comes down to stability, because they have the opposite handedness to all our biological machinery. Mirror image molecules resist being broken down. Our proteases, which chew up proteins, and nucleuses, which chew up DNA and RNA, they just don't recognize the mirror shape. They can't bind to it effectively.
So they just last much longer in the body.
Exactly. Longer half-lives.
Yeah.
Which means you could potentially get sustained therapeutic effects with less frequent dosing.
Yeah.
And maybe even fewer immune reactions, because the immune system might not see them as easily either.
It's like creating stealth molecules almost, alien to our biology. Are there actual examples of this being developed now?
Oh, yes, definitely. There's a technology called spiegelmers. Spiegel means mirror in German. These are mirror image aptamers. Aptamers are like synthetic antibodies, short strands of nucleic acid designed to bind very specifically to a target molecule. Spiegelmers are made from mirror L-nucleic acids. They can bind to disease targets, but because they're mirror image, our natural nucleuses don't degrade them. And some are already in human clinical trials for things like cancer.
That's amazing. Already in trials.
Yeah. And there's also research into using D-peptides, peptides made from right-handed amino acids, as potential new antimicrobials. The hope is they might be less susceptible to existing bacterial resistance mechanisms.
And it's not just medicine, is it? I think the sources mentioned materials science, data storage.
That's right. You could imagine materials with enhanced stability, resistant to biodegradation, or using mirror DNA for incredibly long-lasting, high-density data storage, because it wouldn't break down easily.
Okay, so the benefits of the components are clear. But now the paradox. Why does that same chiral incompatibility make a self-replicating mirror organism so dangerous?
Because it flips from being a benefit resistance to break down to being a catastrophic liability. Total immune evasion. Think about it. Our entire immune system, every part of it, evolved over millions of years to recognize and fight off pathogens made of L-amino acids and D sugars.
It's all geared for our kind of life.
Precisely. So a bacterium made of D amino acids and L sugars, it would be essentially biologically invisible. Antibodies wouldn't bind to its surface proteins. Immune cells like phagocytes, which engulf invaders, wouldn't recognize it as foreign. It would just slip past everything.
Like a ghost in the biological machine.
Exactly. And the consequence would be unchecked replication. You could just grow and grow inside a host without any immune response. Experts have described this as potentially leading to untreatable, pervasive, and lethal infections. Some have called it the ultimate pandemic scenario, because it wouldn't just be resistant to one drug or evade one part of the immune system. It bypasses all of our evolved defenses simultaneously. Nothing like that exists in nature.
That's terrifying. And the threat isn't just to individuals, right? What about ecosystems?
That's the other huge concern. In nature, microbial populations are kept in check by predators, especially viruses called bacteriophages, and by competition with other microbes. But phages rely on specific chiral interactions to infect bacteria. They wouldn't be able to infect mirror bacteria. Other microbes that might prey on bacteria also wouldn't recognize them.
So they'd have no natural enemies.
Effectively none. This makes a mirror microbe the potential perfect invasive species. It could replicate without the usual ecological controls, potentially outcompeting native microbes for resources that aren't chiral.
Like what kind of resources?
Well, simple things like glycerol, acetate, pyruvate, these are common energy sources that don't have handedness. Both natural and mirror life could potentially use them. The fear is that unchecked growth could disrupt fundamental nutrient cycles, nitrogen cycle, carbon cycle that rely on balanced microbial communities. It could cause microbial food webs to collapse, leading to irreversible ecological damage.
And I think the report mentioned photosynthetic mirror organisms as a worst case?
Yes, because a mirror cyanobacterium, for instance, powered just by sunlight, water, and CO2, its growth wouldn't even be limited by needing to find specific food sources. That's seen as a particularly acute threat. It's been described as potentially triggering an ecological singularity, introducing a life form that plays by completely different biological rules, with consequences we can barely predict.
Okay, but let's explore the interaction a bit more. The sources mentioned dietary limits. Mirror bacteria can't eat our L-amino acids or D sugars. Wouldn't that hold them back significantly?
That's a really important point, and it's true. Initially, their diet would be restricted compared to natural bacteria. They couldn't directly consume most of the complex organic matter around us. And some argue this inherent disadvantage means they might not pose such a catastrophic threat. They'd be limited to those acral nutrients we mentioned, or need environments where D amino acids and L sugars are somehow available. But evolution, exactly, that's the counterargument. If a mere organism could survive and replicate at all, even slowly, using just those simple acral molecules, it would start evolving. And over time, there's a real possibility it could evolve new enzymes capable of metabolizing the natural L-amino acids and D sugars that are abundant everywhere. If that happened, the shackles would be off. It could access the vast resources of our biosphere.
So the initial limitation might just be temporary? And what about causing disease? If its toxins are mirror image, would they even work on us?
Another good point. Many bacterial toxins work by interacting specifically with our L-amino acid proteins or D sugar receptors, so a mirror toxin might be ineffective. Similarly, mechanisms for invading our cells might not work due to chiral mismatch.
So it might not be pathogenic in the usual way.
Perhaps not through specific toxins or invasion mechanisms initially, but an organism that replicates completely unchecked inside your body could still cause devastating disease simply by its presence. Imagine something growing uncontrollably, consuming resources like oxygen and simple nutrients, physically crowding out tissues and organs. It could cause systemic failure and death just by its sheer biomass, like a very rapidly growing cancer.
Okay, so the lack of specific virulence factors might not matter if it can just grow without limit.
Precisely. The consensus seems to be that releasing a self-replicating mirror organism would be, as a report puts it, a momentous and irreversible act. It would potentially create a second tree of life on Earth, fundamentally different and with consequences that are incredibly hard, maybe impossible to fully predict, but likely devastating.
Okay, given those stakes, it's no wonder people are calling for caution. Which takes us towards the end here. Part 4 looks at broader implications, and Part 5 tackles governance. What has the scientific community itself said about these risks?
The reaction has actually been quite remarkable and proactive. As we mentioned, that big coalition of 38 leading scientists published that piece in Science late last year, December 2024. And they were very clear. They called for a halt to research specifically aimed at creating self-replicating mirror life forms. They used phrases like unprecedented and overlooked dangers and potential globally disastrous consequences.
That's a very strong and unified message from the core community, and it really highlights the slippery slope problem for governing the research, doesn't it? How do you possibly allow the beneficial work on mirror molecules or components while absolutely preventing anyone from taking that final step to a self-replicating organism?
That is the absolute crux of the governance challenge, because every useful step forward, a better mirror polymerase for diagnostics, a more stable mirror enzyme, even synthesizing the parts of a mirror ribosome for structural studies, each of those incrementally makes the final dangerous step easier, cheaper, and faster for someone else, somewhere else, maybe with different intentions, or even by accident.
So what kind of ideas are being proposed to manage this? How do you draw that line?
Well, several models are being discussed. One is a research moratorium on the specific goal of self-replication, similar to what happened with recombinant DNA at the Asilomar conference back in the 70s, sort of pause to figure things out. Another idea is to define very clear technical decision points or barriers. For instance, maybe research funding and permission is granted up to the point of creating, say, a functional mirror ribosome in a test tube. But there's an absolute hard stop, internationally enforced, before anyone tries to put that ribosome into a system capable of replication, like putting it inside a lipid vesicle with a mirror genome.
Like a clear red line.
Exactly. And another approach focuses on the supply chain, things like rigorously tracking and screening orders for the precursor chemicals, like those expensive L-nucleotides needed for mirror DNA. Similar to how DNA synthesis companies already screen orders for sequences from dangerous pathogens.
Okay, those make sense. But shifting gears slightly before we wrap up governance, how does this whole field intersect with the search for life beyond Earth, this idea of chirality as a biosignature?
Ah, yes, it's deeply connected. Astrobiologists looking for life elsewhere rely on finding biosignatures, signs that can really only be explained by life. In homochirality, a strong preference for one-handedness is considered a potentially very powerful biosignature. Why? Because non-biological processes tend to produce roughly equal mixtures, 50.50 bicyracemic mixtures, of left and right-handed molecules. But life, as we know it, massively amplifies one hand. So finding a significant excess of, say, L-amino acids or D sugars in a Martian soil sample would be incredibly strong evidence for life. Future Mars missions are actually being designed with instruments to detect precisely this.
So if we found, I don't know, D amino acids dominating on Mars or L sugars on Europa, that would basically mean a completely separate origin of life, life 2.0.
It would be incredibly compelling evidence for exactly that. An independent genesis, totally different from Earth life's lineage. It would be one of the biggest discoveries imaginable.
Wow.
Now, there are caveats. We know some non-biological processes, like interaction with polarized light in nebulae, can create slight chiral imbalances in meteorites. And over long geological timescales, chiral signatures can degrade back towards racemic mixtures, a process called races invasion. So you'd want to see a strong chiral signal and ideally combine it with other biosignatures. But the link is clear. Both the lab work building mirror life here and the search for chiral signatures out there are probing the same fundamental question. Was Earth life's choice of handedness just a fluke, a frozen accident, or is there some deeper reason?
And that brings up another wild idea. Could mirror life, or something like it, already exist here on Earth? This shadow biosphere concept.
Yeah, the shadow biosphere hypothesis, often associated with Paul Davies, is this idea that maybe life originated more than once on Earth, and perhaps there are other forms of life here undetected, using different biochemistry. Mirror life would be a perfect candidate for such a shadow biosphere, right? Because all our standard detection methods, based on PCR or culturing with normal nutrients, would completely miss it. It would be biochemically invisible to us.
So how would you even look for it?
Well, the conceptually simple experiment is to take environmental samples, soil, water, whatever, and try to culture them in specifically designed mirror image media. You'd provide D amino acids and L sugars as food, see if anything grows. People have tried this.
Not anything.
So far, no convincing evidence of naturally occurring mirror life has turned up despite quite extensive searches. It seems our type of life really does dominate the planet.
Okay. Beyond the science, though, thinking about actually creating a mirror organism, what does that do to our definition of life itself, philosophically?
It forces some really deep reflections, doesn't it? If we can engineer life based on an opposite molecular blueprint, What does that say about the fundamental nature of life?
Mm.
Is it just chemistry and information? And think about the act itself. If humanity were to create a self-replicating mirror organism, it would arguably be a human-instigated second genesis. That fundamentally changes our relationship with the natural world, doesn't it?
Right.
It puts us in the role of creator in a very profound way.
Yeah, the responsibility is immense. And it makes me think about SETI, the search for extraterrestrial intelligence. You mentioned recognizing life.
Exactly. It's a great analogy for the SETI recognition problem. If we might struggle to even conceive of or detect life that's just a simple mirror image of ourselves,
Hmm.
how much harder would it be to recognize life based on truly alien chemistry? Or to recognize intelligence that manifests in ways completely different from ours? Mirror life makes that abstract challenge feel much more tangible. Our own biological signature is chiral. What if other signatures exist that we aren't even thinking to look for? It really broadens the scope of what life might mean.
Okay, so bringing it back to the immediate governance issue. Given that warning from scientists, what's the current status? Has research actually stopped?
Reports suggest that yes, labs whose explicit long-term goal was creating self-replicating mirror bacteria have largely halted that specific line of research, at least for now. There seems to be a growing international consensus that extreme caution is needed here. But the fundamental challenge remains. The knowledge of how to do it or how to get closer can't be uninvented. And the enabling technologies, better DNA synthesis, better protein engineering, AI for design, they keep advancing, often for completely different beneficial reasons.
So the risk doesn't go away. Rogue states, non-state actors, maybe even just a catastrophic lab accident.
Exactly. Those risks persist and potentially grow as the technology becomes more accessible, which is why the call is for durable global oversight mechanisms, not just a temporary pause. Things like formal international dialogues, maybe frameworks similar to treaties governing nuclear, chemical, or biological weapons. Plus, investing in new kinds of biosurveillance that could detect non-standard biochemistries should something ever escape, and even research into potential countermeasures, however difficult that might be. And fundamentally, it requires embedding a really strong culture of responsibility and awareness of dual-use potential right within the scientific community itself.
It sounds like the community's foresight here has given us a crucial, maybe narrow window to act deliberately, to put guardrails in place before we reach the point of no return. A policy of principled caution, as some have called it.
Hmm.
So wrapping this all up, this deep dive really highlights the stark duality of mirror life, doesn't it? This incredible medical promise on one hand.
And this absolutely unparalleled potential for ecological disaster on the other. It's a perfect example of a powerful technology being a true double-edged sword.
And for all of us listening, it really pushes us to think about the sheer power humanity is developing to reshape biology itself and the enormous responsibility that comes with that power.
Yeah, it forces questions about not just what we can engineer, but what we should engineer and how we make those decisions collectively and wisely.
Definitely. It leaves you with this thought. Maybe the universe is teeming with life, but in forms we just aren't equipped to see yet. Mirror life, whether it stays a tool in the lab or somehow, someday, becomes a reality. It forces us to confront not just what life is here and now, but what life could be. And the really big question, are we ready for that?